well, we love what we do. I mean, let's face it. When you think about endophytes, you really, you have a plant, right? You got to plant beautiful leaves there, and there's something inside. There's something inside those leaves, and that's the endophyte, and that's the secret. That's the secret that this plant harbors. So it is like uh, studying endophytes is kind of like, you know, figuring out what your Christmas presents are under the tree. You got to kind of look inside, you got to open them in there, and you got to study and move them around, even before you open them, try to figure out what's there, and then kind of peek in to see what's there. Then you pull them out, and there they are. There's an endophyte, right? It is. And that goes right down to the mystery of nature, right? The mystery of nature. And that's what, I would argue that we all start out, with that as little babies crawling on the ground, finding a little insect or finding a little plant or whatever it is, or fungus or mushroom, and checking it out. And ended up putting it in our mouth to check it out, that this is it. And this is what this is what carries us on in science, causes us to keep going on. It's because it's basically an extension of what we all do. We all start out this way. And it's just that some of us are lucky enough to be able to keep doing that kind of exploration of nature. And so endophytes. So looking for endophytes is just like a microcosm of that activity, because it is. Every plant is like a gift, and and inside it is a treasure, an endophyte of some sort that has great value, some kind of value, if nothing, just to see it, right? Yeah. So that was interesting discussion that you got started, Matt.
Wow, what a great way to start.
Yeah.
I think we should totally start with that. That was incredible.
Yeah, that was good. That was good. I mean, it's true. It really is true. That's what powers us both, this excitement to learn and to share our knowledge. It's no different. It's we're doing the same thing, really, just exploration. We're
Yeah.
exploring and sharing what we learned and sharing the excitement of it.
It's a wonderful thing. I mean, I feel like at the cusp of a renaissance in science in many ways, because so many people are interested now. So many people want to get involved, and they want it to be understandable and actionable, which is a pragmatism that a lot of, science gets so caught up in these niches, caught up in these individual moments and instances that are not day to day, but people want day to day understanding, day to day interaction, day to day benefits.
Well, the nice thing about regenerative agriculture and and organic and principles of permaculture, right? You get uh, is that there are principles there, and you can see those principles. The same thing with endophytes. There's principles there about how they function, how plants. If we figure out what the principles of how endophytes and plants work together, then there will be simple principles there, so that people can apply that too. Sometimes the details get overwhelming, and you have to step back and see the big picture. Like, that's why I always talk about this. It helps us, this evolutionary biology perspective, right? That's a kind of a developmental biology, a kind of a big picture that you could look at and give you a perspective on how plants work and agriculture in general and what's happened in agriculture. So that is, it's a perspective we don't always have. We all have the capability of it because it's not complicated, but it gives you a perspective on agriculture people don't always have. We're used to our little, we do this experiment, we see this result. And sometimes you can't do experiments. Sometimes you have to kind of look over at nature and compare and do a comparative approach. And it's just as valid, really.
Maybe
Yeah,
more valid.
maybe more valid. Well, you can't do without it because you cannot do, and the other thing that we do, which is good, is we tend to reduce everything, right? Reductionist. This is our modern scientist way, and we've reduced things because we have to in order to test them and break them down and see how they work, right? But when you reduce something to basically, let's say we reduce it down to its DNA, right? You have lost the organism when you do that. Because the organism is more than just the DNA, right? There's ecology there, there's life cycle, there's behavior patterns. And DNA, you break it down to the DNA, you're only looking at this gene or that gene, and it just doesn't tell you about all that development and behavior and stuff like that. So yeah, we break things down, but we lose the character of whatever it is when we do that. And I would argue that is our strength, this human strength, that we're able to do that, and it's our weakness. It's our weakness because we think that's how it is. After we break it down, we think that's how it works. You know, after we into still models. That's the idea. We have models of how everything works, and if something is different than that, we may never see it. We may never know it because we're always referencing our model, our belief system about how things work, which we built mostly based on our reductionist approach.
I
It's
tell
not to
my
make it wrong, but incomplete, right? It's not wrong, it's incomplete. Yeah, go ahead, Matt.
I always tell my students when they ask about like what the hierarchy is, and I always say, well, the morphology of the organism is telling you its adaptation to the environment.
Yeah,
And so the environmental stressors are always going to affect how, I mean, evolution has shown us that things develop fins over and over again, right? To be in the
yeah.
ocean.
Oh yeah.
And it's that common adaptation to the stressors of the environment that is so key to understand because some of these microbes can exist in huge pH swings, can exist in huge EH swings, and they change form. They're pleomorphic. And so, sometimes they look like false fungi and sometimes they're bacilli. And it's in that expression that tells you the environment.
This concept called the habitat adapted symbiosis. And this was proposed, I think it was around 2014, somewhere in that time, by Rusty Rodriguez and Regina Redmond. And they were looking at how plants were associating with fungi, with endophytes, were breaking in, they were absorbing endophytes from the environment as they went down a climb to a high salt soil, right? And they noticed that their fungus, their endophytes were changing in the plant. And they later on isolated the endophyte out. It turned out it was a fusarium, but they could put it in other plants and it would make those plants salt tolerant. This, so the concept is that plants are creating symbiosis or endophyte symbiosis, right? Endophyte taking in endophytes that enable them to adapt to that high salt stress environment. So the same thing for any environment. The concept, you see the same thing with heavy metals, and that is plants will in those heavy metals will take in certain endophytes, and that enables them to survive in those environments. We don't really know what's happening in the plant, to be honest. We don't really know. We have hypotheses, right? We have hypotheses and ideas. They're pretty smart ideas, I think. We made one of those ideas. We call them smart, but it has to do with antioxidants. And that's because with endophytes, plants take in endophytes and they that causes them to become more resistant to oxidative stress. They produce more antioxidants internally and chemicals like phenolics that are antioxidant properties. And so stresses tend to be translated into oxidative stress, right? So whatever you have, heavy metal, lead, or heat, or whatever it is, it turns into ROS, reactive oxygen, and then that's oxidative stress. That's oxidative stress, and that then damages the plant's DNA. So if plants are already, because they have endophytes, they're already full of antioxidants, right? Then this reactive oxygen doesn't hurt the plant, doesn't cause any damage to the plant. So that's one idea. But there's other ideas about that. Why endophytes give plants, and there's another idea is, and you can't say one is more valid than the other. The other idea is that the endophytes themselves are producing substances that are protective of the plant. And I mean, we could think of I don't know, it could be beta, let's see, uh, uh, who knows, carbohydrates, some carbohydrates that the fungus could produce, or if they're bacteria, it could be organic acids, that have some protective effects in the plant. Any compounds like that, right? That we don't know. There's any kind of interactions that could be happening that have effects of protection on the plant cells. So our ideas about what's happening, they're ideas with basis, right? With basis, but that's what they are. They're our best knowledge at this point.
We're getting to the point where folks are understanding that we have to allow the plants really to speak to us, allow the biology to speak to us, because in taking a non-definitive stance is something I really try to instill in my students. Even when we have a positive ID, you got the thing right there. It's like, yeah, but things can morphologically look alike in the jungle, so why wouldn't it be here on our slide? So we got to keep it non-definitive. And even, it's we're developing techniques to try to individually grab that spore, or I mean, some spores are huge and you can do this, some spores are
Very
small.
hard. That's
So
very
hard.
hard. Those
But
tiny things. You can get micro manipulators,
yeah,
but they're hard to use. I mean, you could get different kinds. You put some kinds, you hook them up with your microscope, and they're still hard to use. It's very hard. Really, there are old ways to do it that work
the pipette breaking.
best. Pipette breaking. Did we
Yeah.
talk about that? You figured that out, right? You did that.
Well,
That's an old method. That's an old method.
yeah.
Tiny, 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 tiny little hypodermic needle, right? Like
Mhm.
that. Pull your little capillary glass out, and it gets really, really tiny, like a little syringe, right? And then you take it and you look at it in the microscope, and you get it close to the spore, and it just, with capillary action, it soaks it up. Then you have to take it and blow it out, or break the tip and immerse it, and hope it'll grow out of the glass.
Yeah.
Yeah, I'm sorry for interrupting, Matt. You go on. You were saying
Oh, I was
something.
just saying it's so much work. It's so difficult to isolate these things to really know what that one thing is. But it doesn't quite matter because the reason it looks that way is because of the environment. And if we're dealing with the environment, the soil environment, and trying to understand it holistically, it works perfectly. We don't need to know precisely what that individual thing is. I like DNA. We're gonna have a lot of DNA speakers that are gonna join us. I love the grounding it gives us. The fact that people are like, oh wow, it's so diverse. I didn't know it was this diverse. Oh, it can have those microbes. Wow, like that kind of grounding and also myth busting. What I really look forward to is the day where we can take a cubic yard of soil, 10 centimeters deep, and every single bit of it we DNA test and keep track of where we took it from. So we'd have to bit by bit break it off and then send that through a series of tests, but have it be biologically, visually, microscopy tested, but also DNA tested and mineral tested, and have that all mapped out so that we could fly through the microscopic world, see it all visually, and then and click on things and know what they are. And I would love that because I think that there's a massive amount of variation within that cubic yard. And it's never, people don't understand and don't consider how big of a variation there actually is. And I think that would ground us in a unique way. And that's so much of my work is like grounding us.
Well, I think that's right. I think not only that, it is in essence we are pardon me, seekers of truth, right? Seekers of truth. So you really, this is an area of human exploration that gives us a tremendous amount of information, metagenomics and the study of genomics. It's not everything. I mean, it is a reductionist, but really metagenomics is less of a reductionist. It's more comparable to what we used to be doing when we make surveys and just isolate stuff out, right? More
Right.
comparable to that, but we need to know what's in there. I mean, and you may need to know on doing experiments like like we're trying to move microbes from like about our project to try to explore the microbiology of this landrace corn, right? This nitrogen efficient corn, right? You
Yeah.
gave us corn to grow, so we're working on that, and what we're trying to do is to move the microbes from without having to isolate them, right? Because you can't isolate most microbes. You can isolate some. So we're working on some like maybe I don't know. I've heard different numbers, but I've heard 1%, only 1% can be cultured of species. So mostly, oh my God, most things you can't culture. So what we're trying to do is we're exploring ways to move endophytes from one plant to another plant without culturing them, right? That's the idea. So that potentially we can um, improve our crop cultivars by putting microbes back in those cultivars from, say, wild relatives, right, of those crops, right? So fix our cultivars that are damaged, our damaged crops. Okay, that's the theory there. But to do those experiments, we can we can get microbes from one and put it in the other, but you really have to have, and this was my whole point of bringing this up, you really have to have DNA, that metagenomics, to make sure you move those microbes over, because I mean, we can look in there and we can see some things in there, some bacteria in the cells, but we don't know what they are, whether we move them over, right? So we have to have DNA. I mean, so my point is, it's really critical to have that kind of data, right?
Absolutely. I think it's like a summative check, but the progressive checks must always, and the summative check must be also checked with the microscope. I feel
Yeah,
like the microscope is the living context.
I
What,
did too.
what, how much of the microbes that we're looking at can become in a no-till regenerative ag system regenerative? Like every year, like rhizobia, you put it in once, it's there. Mycorrhizal fungi, if you respect the soil and you're constantly putting, you put in a cover crop instead of leaving it bare,
Mhm.
and you avoid phosphate, as fertilizer, it's regenerative too. I wonder how much is missing, and I wonder how much we can just bring back and have it stick and not be every year we
Well,
add this.
I think the key is it's all regenerative if you keep the soil alive, right? I think
Yeah.
that's the key. Keep it alive. And what does that mean? That means don't dry it out for a long period of time. Don't do deep turning with the plow. Keep living plants, some kind of plants, a cover crop, right? The more the merrier, as they say, 10 or 20 species, a mixed species cover crop, if you can do that, so keep the soil alive. That's the key right there. And then keep building it, keep improving it. Continual improvement, can't beat it.
It's an exciting time. I think that we're going to see a lot of these things become well known and accepted in mainstream. I think we're at a cusp moment in history where people are going to become microbially aware. And people are going to recognize that even if we sometimes can't trace the exact exchange, the exact process, the exact mechanism, that having these microbes there, we have provable basis. We've shown it. It happens in all the different bioregions, and that's exactly why I want to do this database, because I want to let it play out, let it speak for itself, because that's what I want. I want it to, the lactic acid bacteria applications on corn, to be
Yeah.
replicated everywhere, but not just like a university did it. You know, it should be like Joe Farmer, Susie Farmer, and everyone's done it in the bioregion and tried it on a test plot, and they're like, this works here.
Do you know Joe Farmer, by
I
the way?
do.
Yeah, you really do. Yeah, he's a nice guy. I'm not joking. You know Joe Farmer actually? He's a person's name. It's a person's name.
Oh
Actual name.
no.
Yeah. Yeah, yeah, yeah. No, I know a Joe Farmer. Yeah. He's a really nice guy.
That's
Really,
awesome. I love
really,
that.
yeah, really good guy. Yeah, no, it's the the point you were making is really good. And what was that point, Matt?
Ah,
What was
that
that point? What was
my
that
point was is that
before I
that
interrupt you?
that all these folks are going to be able to see these things happen and let them play out. And
With
when we
microscopy, right?
right, and also
Yeah.
testing. I mean, it's
That's
not just
why I love.
microscopy; it's
Oh
pH.
yeah,
When
yeah,
we
yeah,
can look at the
yeah,
microbes in relation to
yeah.
pH across bioregions, woo, it's going to be wild.
Well, I think that's one of the really important things, and this kind of goes along with the microscopy movement, right? But the really important things are that so that people can see how plants use these microbes themselves, right? Because they
Yeah,
can see these microbes in soils, see them there, see them, and so they can evaluate for themselves some of the stuff that people are saying, about mycorrhizae and about bacteria on roots and in roots, and they can see that themselves. So that's the important. I think that's an important empowerment, right? Because we really are talking about with regenerative agriculture and permaculture, organic farming, we're talking about a people's movement, and it's powered not by industry and big money and even universities pushing it. It's powered by people, right? People drive this movement, and they need, in order to do that, they need confidence to know how things are working, right? And
yeah,
when they see it with their own eyes, they don't listen to me. They don't have to listen to me. They don't have to listen to you, or 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 other people, talking about plants and microbes or soil microbes. They can look at it themselves. That's more important, I think, than any test that they can do,
yeah,
because that that puts the power in the person who has to have the courage to do regenerative agriculture, right? They have to have the courage. And so they have to have the certainty of knowing what they saw, right? It's a human thing. It's a human thing. That way they can't be hoodwinked by any company that comes after and tells them this or that, right? Or somebody else that denies, oh, there's no microbes in those plants, or there something along those lines. It's all chemistry. So they have to, in seeing, I mean, like they say, seeing is believing, right? Seeing is believing. it's at least a step, right? It's at least a step. You don't have to take other people's word for it. You can put your own eyes on it and see it. And that's to me, I think that's the most important thing that we are doing, really.
I completely agree. And because it's a value and a principle that transcends not just what we're doing, it transcends the whole regenerative agriculture movement, all of science. We want it to become, we want it to have the ability to be self-evident so that people can prove it to themselves and build confidence and stand. I always say, like, thin ice, thick ice, you know what I mean? It's stable ground. And there's a lot of things that people just assume are thin ice and they don't, it's because they can't test it. But once they bring it into that human modality, it changes everything. That's why I started doing stains for numeracy. So the biology, like, it soaks up acridine orange when you do it super diluted. You just put like 2 drops in 1 ml of 1 to 100 water to soil the water, and they soak it up in 2 minutes and they glow. Or if there's no life in there, the whole slide is like a fog of glow. And so it's like, well, you actually can see it as a gradient of that. And so all the bacteria that you can't see because light microscopy doesn't go far enough, you know, we can actually gather in this, just, this gestalt test. So simple, so easy. But at the same time, the farmer can be like, yeah, I can see that your work with me on this site, this consultant had, we've done this compost, we've done this biological amendment. Maybe we're putting just EM in the drip. They'll be able to see the difference in the biology numbers. They'll be able to see the difference in the soil organic matter, in the pH. All of it will correlate, but it will find its home in the visual. You'll be able to see it. It won't be just a number. It won't just be a color. You'll be able to see it.
Cool. That'll be cool. Very nice.
Well,
Yeah.
I think of you. I
Yeah.
mean, you've got. I always tell people it's like I feel like you're like the chef or the king of stains when you've created all these amazing solutions to be able to see these different behaviors, different mechanisms and processes. And for me, I always want to cut to like the simplest thing, and that's why I did blue methylene on the root hairs to just show the end of the process, because I want the farmer to just be like, "Oh, I can do that. That's it. I can do that." And it's
Well,
that
no,
connection.
you're
But
right. You're disdains for different functions, right? Stains for different functions.
yeah.
And that was a function, right? You wanted it simple enough that it's not going to be overcomplicated for others to do, right? For other people to do. Yeah. And my function was different. Don't
Oh yeah,
forget.
I know.
It
It was
was very
critical.
different. I mean, here we have these things inside cells. So how do we see them? And how do we show they're actually bacteria, right? So they're actually microbes. So then you go in, what are they producing? Are they producing, what hormones are they producing? And so you screen, you look for something, okay, for ethylene. Okay, oh, we got ethylene. What else are they doing? Are they making nitrogen? I mean, so then we start looking for things that stain nitrogen. And there's nothing there for microscopy, for looking at microscopy. So these things are experimental really experimental, right? So you take other kinds of chemistry and you kind of bring them together and adapt them for work on microscopy tissue, and then then you try to use them, and
Do you study medical journals too?
you go to the medical journals because see the medical see don't forget people in medicine. I mean, we've been putting so much money in medicine for such a long time. So sometimes, some of the best basic information is in that medical literature. The next is the plant literature. At the bottom is the fungal literature.
And that's
I
the
can tell
truth.
you that.
That's
That's
how
the
that's how I
truth.
figured out the glow
Yeah,
for
that's
the
the truth.
roots.
Yeah,
The
that's how it
myco
is.
the mycorrhizal glow in the roots. It's very difficult to find anything in the mycology world, but as soon as you figure out that that's NADPH, it's an NADPH form of phosphate. Oh, the medical literature goes back 40 years on how that's a biological autofluorescent biomarker that they use. So is NAD plus and NADH plus. They glow too, and that's also here. So
Cool.
it's a precursor. But what's wild is it's the NADH plus is part of nitrate to nitrite, and then that's the precursor to forming a phosphate. So it's like all these cycles are all linked. A lot of people are like, I need more of this, I need more of that. I'm like, well, they're actually all working together. But I had to go to the medical, and it's oh man, that's so great that to hear you say that because I've had to do that with a lot of things to figure things out.
I do it all the time. I do it all the time because you get such detail there, for, I mean, we can think about it with human health. We've put so much money into human health over the years. So that's well studied. And even when you think about the animal, you think about animals, if you think about when you go to college and you study biology, right? You get a lot on animal biology and very little on plant biology, and nothing on fungal. It's thrown in with the plant
Mhm.
biology, right? So you really, and that may be the end of it, unless you're like a botany major or plant biology major or something like that. That may be it for if you're a biology major. You're going to get a lot of animal stuff and medical stuff, and that's going to be it. We put all of our focus on ourselves.
Well, and there's a massive reaction. I mean, America and all over the world, there's a huge wave of citizen science in mycology right now because they just
Yeah.
didn't provide
Yeah.
for it. And so people are hungry for it and they're going after it.
Well, fungi are one of these groups of organisms that there's this mystique, right? It's kind of people don't know very much about them, and they're poison ones, and they look really cool, you know. Maybe have a dangerous kind of feel to them, kind of a reputation. So that kind of makes people curious, and especially modern people. There's a younger people today. There's a big curiosity in fungi right now. So my mycology classes are full, and we this semester. In fact, this semester has been a good one has been a challenge for teaching mycology because we're in a drought now. So we really have to hunt for
Oh.
fungi. Yeah, and around New Jersey right now, it's a drought. It's a drought. We haven't had very much rain in at least 2 months, and that's suppressed all the ground mushrooms, and the wood mushrooms have popped up. They've continued to pop up, but a, a part of the fungal community is gone because of the dryness. The ground is so dry.
Maybe a field trip?
We have field trips every single week. Oh, do we go? We go in the woods every single week to hunt fungi. But the my problem is, yeah, a field trip is a good idea. The problem is there are practical constraints, and like I would have to get a bus to carry everybody, right? To carry everybody. So that's a constraint. Too many students. So really, we're locked here. We're okay now because the semester's over and we have some other things coming up that'll finish out the semester. But if we have another year like this, I'm going to have to rethink how we teach mycology. And this is
Yeah.
the best
Do you
way.
bring in like all like the koji and all the miso and that whole world? Because
We talk about that, but actually we go into the woods is what we do. We go in. So I give lectures. I have recorded lectures that they're supposed to listen to. I think probably most of them don't listen to it. And then, and then when we meet, it's supposed to be a review, but I cover everything really. So I go over basically what was in the lecture. It's not very efficient, you see. But the idea is to learn, right? To learn, to give them the practical. That's one day. And the other day, we go in the woods. I take the students in the woods We meet, we spend about 80 to 90 minutes hunting mushrooms in the ecological Rutgers Ecological Preserve. Some woodlands we have, a natural area that we have as a study site, just the woods basically. And we go hunting mushrooms and studying fungi. That's what we do. And if it's dry like this and we can't get the ground ones, we get only the ones on the trees. Now it turns out it's enough because we've got pretty good amount of variability. And the students are also going to some other sites where they may have some other fungi. So it worked out okay, but many more years like this, and I don't know what we're going to do.
Mm.
Well, I always felt that microscopy, so has potential for being an art form. Really, when you think of the microscope images, and if you think of earlier biologist Ernst Haeckel. Matt, do you remember who Ernst Haeckel is? Ernst Haeckel. Okay,
He's
Ernst
not the guy who did the first microanimals, right?
Yeah, he did. He did. I don't think
Okay.
it's the first one, but Ernst Haeckel was a German scientist, 1800s that into the 1900s that uh, he developed this idea called uh, was it ontogeny follows phylogeny, something like that. It's basically it's the idea that organisms develop through their evolutionary stages. So a human, for example, a human embryo, if you look at the way it develops, there's a stage early on that looks like a little fish, right? Looks like a fish embryo, kind of looks like a little fish, right? So they say, so the idea being that represents that evolutionary stage of development as animals evolved, right?
Hmm.
This idea of, and it's said different ways, but ontogeny or development recapitulates or tracks essentially phylogeny or evolution, right? That development stages track how organisms evolve. Okay, so that was his idea. And so it was an early on idea of evolution. I don't think it's valid, but I mean it's still you do see examples like that where you see uh, developmental stages that look like other simpler groups of organisms. That's absolutely true.
I think it's pretty fascinating that, when we talk about animals in the ocean, talk about organisms in the ocean, so many of them start off microscopic, but people don't think about the fact that the human experience starts microscopically. If you abuse a pregnant mother, you can damage the child for life. So the experience starts in there. And so we have some sense of this other world, this other existence microscopically, and we all disintegrate at the end of our lives back into the microscopic world. And so there's this profound understanding that this is a space that is mysterious, but we're also drawn to it. We're also familiar with it. We have this understanding that this is a touchstone for all life. This is a cyclical node in all existence. And and that's, I think, part of the reason why we're attracted to it. It's part of us in a very fundamental way.
Yeah, yeah, I think that's right. The this is a this is a oh hell, man, what were we talking about now? What did you just say? A more fundamental way. Cyclic, we're part of that. We're part of this cycle, right? Part of these natural cycles. Okay, there's no doubt about that. And it is fundamental. I mean, to know that we're part of those cycles is,
We
I mean,
sense
that's,
it. But that's
yeah.
what we're essentially grounding when we look with the microscope, when we do tests and get
Oh,
feedback,
yeah.
we're
Oh, yeah.
grounding ourselves into
We
that natural
are,
system.
we are, that's a perspective that I haven't dwelled on very much, but it's true. I mean, it's more like recycling energy, right? The energy of what our bodies are, going into this microbiology and into other organisms, into the inanimate, and then cycling through again. But of course, what we lack there with that idea is we lack our consciousness moving with that energy, with those energies. Yeah. So yeah, there's no doubt about it. I think more about the cycles, Matt, are we're attracted to cycles.
Mm.
I think we're attracted to cyclic things that are, nutrient cycles, for example, to understand them and see how they move through the system and how they're generated and stuff like that. I think it's a natural thing that we are just naturally.
Because it's within us. We are a physical manifestation of hierarchy of stacked cycles. And so we see those cycles in ourselves. I think that's why we're
Yeah.
attracted to it.
Yeah. So it's very basic, very primitive that we notice those cycles. I'm thinking of the primitive symbols of, I mean, what about the Buddha symbol, where you have this snake that goes around or this thing that goes around, right? A cycle there. And I've seen cycles on Aboriginal rocks, right? Where they do a cycle, a spiral, and the cycle, uh, the Stonehenge cycles, right? That is, we make these cycles. I think we have a fixation on cycles, on cycles, wherever it comes from. I'm sure it comes from, it's very basic about us because, I think it is this. I think it is this because we see day night cycles, right? We learned, year cycles and lunar cycles. And so people got a fixation on those cycles early on and really, so now we look for cycles. I guess it's also logical, if you have a finite something, how does it move in the environment? How does it come back? How does it move around? So it's a logical thing. So yeah, it is interesting. Our fascination, fixation on cycles.
When I first started on regenerative soil, I was surprised at how little visuals there were on so many of the mineral cycles. I feel like I had the great opportunity to create those because no one had created them. And it does feel like there's a hunger for cycles, but it also feels like we don't know most of the cycles. Like
Yeah.
when I learned about like all these dominant endophytes, like with Saccharomyces cerevisiae, lactic acid bacteria, purple non-silver bacteria, Rhodocytomonas palustris, and then I find that these are in all our probiotics. Oh, these are the things that are helping people with their gut digestion. I'm like, well, how common are these to the gut? I'm like, oh wait, this is the feedback loop between our world, and then everyone's like, eat local. It's like, well, yeah, you're getting into the actual feedback loop of the microbes for your actual bioregion. And it's profound, but it's also so simple.
It is simple. It is simple. It really is simple. And cycle, we'll mention one cycle, okay, the one that we published, right? Rhizophagy cycle, right? Rhizophagy cycle, the root eating cycle, where roots are attracting microbes to the tip, absorbing them into the root cells and hitting them with reactive oxygen, extracting out the nutrients, and then replicating the microbes and putting them out on the root hairs, putting them out from the tips of root hairs. So essentially ejecting them back into the soil as those root hairs grow. Okay, so that's the rhizophagy cycle. And the plant is getting nutrients from the microbes that it's absorbing oxidatively, oxidizing them with superoxide to get the nutrients, oxidizes the walls off, it takes the nutrients, and then ejects the microbes that survive out the tips of the root hairs, and they go back to the soil, get more nutrients, and later go back to the root tip following exudates. So that's the idea of the cycle. So it's a rhizophagy cycle. It's a plant cycle. It's a microbe cycle, right? It's a plant microbe cycle. It's essentially cycling microbes between the soil and inside the plant and getting nutrients from them and putting them back out into the soil. And that's what's working in the on the roots, on the root, down in the tips of the roots. And so that's one cycle. So it was actually, I think, Matt, that people were excited about rhizophagy cycle. I think part of it is just because it's a cycle, because it was defined as a cycle, right? That
Well,
makes it.
it actually redefined everything from the permaculture folks. So maybe I never explained this from our perspective to you, but everyone in permaculture was told like, oh, you need to add compost tea. It's gonna, do all these different things, and it's gonna cause the nematodes and the protozoa to have manures, and those manures are the thing that feeds your plant. And it was never like, oh, your plants are just going to directly feed on it. But they figured out in the process of doing this, they're like, oh, the best compost tea application happens at the root, the growing root tips of your plants. And it's like they proved rhizophagy with compost tea as a practice as what was best and most effective, but they didn't have the mechanics of why it was effective. And so when I was essentially retelling the story of compost tea to my students, I was like, this is the real reason why compost tea is working so well. It's not that you're immediately getting microbial manures immediately right there, happenstance. Those things generate over seasons and over time where there's a buildup and accrual of microbial manures, and then the plant's traveling through that, taking those things out in the space of absorption of nutrients. But this is fundamentally different. This also is it even if you don't have nematodes and protozoa, this happens. So the idea that there is a hierarchy, the idea that there is a natural progression, that this was also logically part of establishment to permaculture people, this is wildly important because for the longest time they've thought that these other paradigms and mechanisms were at work, but this explains why the things were working the way they were. And so for me it was like, oh, and then when I told people, they were just like, oh my gosh, yes. And so that's from my perspective in my world, that's why rhizophagy took off like wildfire, because we had the pieces around the idea that you
Yeah,
brought and articulated to the community, and it just lit up and also gave people more confidence because it explained the why of what they were seeing.
yeah. Well, I really think that, we have, how research develops. You know, if you think a certain thing, like you think that plants work with chemistry, then you develop a huge chemical, product inventory of how to and methods of how you manage plants, using chemistry, right? But if you think that biology is important, right? We don't have very many people disseminating knowledge about and exploring, not much money, not much explore. I mean, comparatively,
Mm.
right? It's basically nothing comparatively going into, how you grow plants using microbiology or organically, or so uh, there's an absence of research, right? So this research that we have done, knowledge about rhizophagy and working on the endophyte area, right? The endophyte area is and that's a huge area, really. Endophyte area, not all doing what we're doing, but really fits as permaculture science and regenerative agriculture science. organic agriculture science. It really fits there, you know. And it's that, I mean, I think it's a part of of the of the theory, right, of the field, right? Becomes a part of the theory or the knowledge or the hypotheses, let's put it, about how it works, right? And that was absent. That was absent. And now there's a piece of biology there, a piece of science that you can then refer to. Helps explain also. I think, and that was, casual, that Matt, I mean, it was just pure accident that we ended up with that, honestly, just people, studying how microbes, studying endophytes in plants. That's it. And you think, well, what could be less impactful, right? Because we don't even want to do science that has big impact. We want to be left alone. Just here, give us a microscope. Here, get some stains here. Let's go. Come on, let's see what we can find in here. Let's look at this and see what we can find. Oh, come on, let's write a little paper on that. You get the picture? But by accident, that was turned out being an important paper, right? And it just so happened to be some science that really belongs in regenerative agriculture and permaculture and organic growing. So it just so happens to be important, but it's not because we set out to do that, something that was important.
But that to me is like that is the thing that I see throughout all the history of science. You've got methylene blue being used for blue genes, and
Mm-hmm.
suddenly it's now a very popular metabolic drug that's very cheap, and people are using it for all sorts of things. And it's also our stain. But there's so much of that unintended uh, benefit in science. And there's also unintended harm, of course. But I think if we have our eyes open and we have community transparency, those things could be detected and avoided instead of capitalized upon, as we often see.
Well, here's the scary thing, Matt, and that is that we have so much noise out in science
Hmm.
and everywhere in society, right? Everywhere we're online, everything is online. You got noise, this, that, people doing this, that, scientists following this grant program, that program, news happening constantly, right? So if somebody does, let's say they did in fact, this is how most science happens. They do some work and they publish it and they get the pub accepted and published. It goes into a journal and it may lay dormant there for years, forever. Let's face it, forever. It may lay dormant there forever, forever. Yeah, the the thing that one has to do, what I've learned, is if it's something that you think is important, then you have to do like companion papers, right? You have to release the science paper, publish that, and then simultaneously release a paper, have a university reporter write an informational paper, a little article about it, and then release that a press release, so that we see those all the time, these little science reports. Well, that's how those things get generated, as the investigators will say, well, we think we got something interesting here, and we think it'd be worth putting a press release out there. So then you get a little short article of some sensational piece of science, right? That's how that works. And those usually will point back to the article, right? And as it so happens that Rhizophagy article was like that. And one may never have seen it otherwise because it didn't have that press release thing on it.
Is
Yeah.
that where
So,
John Kemp saw it?
well, everybody saw it because
Yeah.
it had a press release with it. Yeah. So everybody saw it. A lot of people saw, oh, that one. Oh my gosh, Matt. I was in shock, literally in shock, because we can count when people are downloading articles, right? And for like 3 days, the download the download was like going up like this, more and more and more and more people. It was really, really, they were downloading like like every second, like every second it was being downloaded. Every second it was being downloaded. That went on for 2 or 3 days like that. And then eventually went down to like 15, one every 15 minutes. But it was, I never saw anything like that.
It's amazing.
That paper, how many people were downloading it? It was amazing. It was amazing.
Well,
Never
it
any
is.
research before, never any research after like that.
Well, I think the your work with trichomes, I would expect that to get picked up.
Not as much. No, not as much. Not as much. Why is that? I think that's because rhizophagy is so much more relevant, I
Hmm.
think, to people because it's right in the soil, it's right there. It's a process that's happening in the soil. Trichomes are up in the plant. Yes, there's nitrogen going there. There could be other nutrients coming out of the microbes there, but most of the work is happening down in the soil, right?
Yeah.
It That's the only thing that makes sense. And everybody knows it. And everybody knows it. And everybody knows it. And that's why that paper is of more interest than the trichome paper. It's because that's the most important place for this to be happening.
It's so exciting. I I mean, man, it's so exciting. And I've seen that you're speaking now to more and more farmers at farmers conferences. The word's getting out. It, we need those microbes. We need them to be cycling. It's the only way that plant root hairs develop, and it's good. It's the I think the fastest pathway to lowering our inputs. Especially in terms of nitrogen.
I do too. Oh well, phosphorus too. I mean, look. I mean, if you take the whole thing as a package, right?
Hmm.
You've got soil health, work on soil health, improve soil health. You're improving your soil microbiology, and you could improve then, and then part of that microbiology is cover crops, right? Cover crops, getting microbes back in there, and then growing your plants there, growing your crops there. So it's a whole package. It's endophytes. You might use compost teas, whatever you're using. Whatever you're using.
Mycorrhizal
Putting
fungi.
mycorrhizae in there, putting mycorrhizal fungi in there, and it's the whole thing. Soil improvement and adding microbes, microbiology. Yeah,
I think we're really at the cusp of massive adoption because we have the ability for the first time to portably show the mycorrhizal fungi inoculation was successful. We can now have my students go, or anyone with this microscope and my book can go out to a field for any farmer and check their roots and be like, listen, your mycorrhizal inoculant was 80 to 100% successful. I checked every 1,000 plants in this 1 acre. It's all, and then you show up next year and just verify that it's regenerative. Don't need to do anything else. And when that happens, when that adoption happens, I mean, some of the literature, it's highly variable depending on other soil conditions, other microbes that are present, but it can be up to 30, up to 40% increase in yield. It can be down to like 12% too, depending on all those other factors. But it's if we just have mycorrhizal fungi, we just have the rhizophagy cycle start again and be healthy. Because there's always, I mean, it's not just, oh, is it happening? Is it happening at the rate and at the level that gives this plant what they need? When we reach all those sweet spots, I mean, we're going to have so much more yield on the same plot of land. We're going to be improving the soil, so next year is going to be easier, and there's just going to be this 10 to 20-year span of absolute change, sea change for the experience for the farmer. And then the end result, and it won't be that long into the process, we're going to see really good food. And then food is going to get better and better beyond expectation, beyond anything we've ever tested, because we've never done all the variables to the capacity or ideal proportion. So I'm so hopeful for the future in this regard.
I think the key really is to support the movement, right? Support
Yeah.
the move, support the, regenerative and organic. Just keep it from my perspective. That's what I'm trying to do, right? With as much as I can, with science where we can, and with the talks where I can. The problem with doing too many talks is I have to travel, and that costs so much. And I have, I'm a professor here, so I have a full-time job here with graduate students. So it's hard for me to travel. So I've been trying to make as many online as I can, you know, because that
Yeah.
is, it just costs so much time, and time is, that's critical. That's
Yeah.
critical. And it wears you out when you're just traveling all the time. Uh, I miss being home with my wife, right?
Yeah.
I feel guilty about not being with my students, taking care of my students. You, you see, yeah. I try to make them online as much as I can. It's just I don't want to. I love my job, Matt. I love my job, and I don't want to turn it into something I don't like, which way over. You know what I mean? I got to keep it manageable so it's not killing me.
Mm.
Provide the support where I can, but not, uh, but not go overboard because
Yeah, we need you to keep making discoveries
yeah, got to
and it's
balance, got to
yeah,
balance.
and I love your papers because of how much you include your students and honor them. They're not there's names at the top. You've got their pictures and then you credit who took the picture. It's
Yeah,
I mean, you're lifting these kids,
yeah,
and to me,
oh
they're
yeah,
they're the same age as my students.
yeah,
They're my students. My oldest
yeah,
students now are
yeah,
27, 28, my
yeah,
graduates,
yeah,
and it's so critical to provide that opportunity for growth, recognition. It's a true hallmark
yeah.
of an amazing teacher.
Well, that's my role, is to support them. That's one of my roles, and our role as this research program is a team effort, so they're contributing to it. So I always give them credit because of that. And I need their support too. I want them. I mean, they're the first. If I see something new, or if we have something new, then we want to make sure everyone sees it and thinks it's right, right? I mean, this is the evidence. This is essentially when we write an article like that, it's a testament that that's what we see, that's what we found, right? So we're essentially, swearing to it. That's what we found right there. This is it. Yeah. So everyone should be on board with that. So it's the first test is who writes it. So and it is. I mean, we're all equals. We're all equals. We're all equals. And actually, they're getting more equal as I get older. I'm losing brain cells. It's true. I'm losing brain cells, man. It's coming. I'm almost 66 now. I never was supposed to even be alive at this age. Wasn't ever supposed to make it.
Well, I'm certainly glad that you're here.
Well,
My dad's considerably older than you, and he's going to start on lion's mane.
yeah.
I don't know if
So
you.
that's what I should do. I thought about that. I thought about that. Thought about trying it.
Well, it's
Well,
pretty
we
wild. I
collected
mean,
some this semester. We collected some, even though the drought was going. We found some lion's mane growing this fall out at the Rutgers Ecological Preserve.
that's beautiful. The literature on the neuro regeneration and
Yeah,
the arresting of neurodegeneration by lion's mane is compelling.
yeah, yeah. I'll pick some up. I'll pick some. Yeah,
For
I'll
all
pick
of
some
us.
up this weekend, or we'll go find some. We still might be able to find some. They're wood decay, so there's moisture in the wood, so they're probably find some out there if we look around. If we go far enough out, we're going out this afternoon in a few minutes. I have a class of students, so we're going to go here on campus and go to another woods, and because they have fungi, they have to find for their class assignment.
Beautiful.
So yeah, so we're going to go out there and look, see if we can find them, see if I can help them find them.
Wonderful.
Yeah. Yeah.
So how much time do you actually have?
Yeah, it's about time.
Okay.
We're about there, I think.
All
I'm
right.
getting tired. I don't
I
have the
appreciate
energy
you.
you have, Matt. I don't have that kind of energy.
I don't always either. I do a lot of meditation and breath work. I do yoga. I do qigong. I read
Yeah.
books that kind of pull me out of the stress of the moment
I'm
to
meditating. I've been doing meditating too.
really.
Yeah, I've been doing meditation a couple times a day. Yeah, a couple
Mm.
times a day. And that it makes a big difference. Makes a big difference. Yeah. To the managing stress. And you're right. Stress is that's the idea, is keep the stress level low. Yeah, that's the idea. As
That is
much as
the idea.
possible, right? As much as possible. Yeah.
I love
And
it.
it's more important when you're older too. It's okay, when hell, 5 years ago or 10 years ago, I could handle a lot of stress, and just manage it and not worry about it. But now it really gets to me,
Mm.
stress, so the capacity to tolerate stress reduces when you age.
Well, I will be meditating more
Yeah.
then.
Yeah. 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 It helps too. It really helps. It really helps. The meditation really helps. Absolutely.
Well, we appreciate you so much. I always learn, and this was so much fun to go back and forth on ideas, and always appreciate you sharing.
Thank you, Matt. I enjoyed it. Thank you. And I really appreciate what you're doing, everything you're doing. Yeah, I appreciate it. Thank you.
All right, it's finally happened. I've been working on this project for years, and I want to invite you to it. It's a free online conference. It is Our Soil 2024. This is our first annual one. We're going to be doing it every single year. It's free. We have over 30 speakers. It's 5 days, free replays, panels, amazing giveaways, and we're going to be doing live Q&A so you can get your questions answered. You can get things figured out for you. Join us. It's going to be a very unique and amazing experience. I'm Matt Powers. I'll be hosting it. Grow abundantly, learn daily, and live regeneratively. I'll see you there.